Hi everyone, this is Rodolphe Sossou Tchatcha. My mentor and I work this semester has based on the application of antifreeze proteins in cryomedicine and in the treatment of frostbite. And we should ask ourselves, how do Antarctic fishes survive despite the freezing point? It is simply because they have what we call antifreeze proteins. Antifreeze proteins have cryoprotecting role. They bind to crystals and inhibit the ice recrystallizations. They lower the freezing point by creating a big gap between the freezing point and the melting point called thermal hysteresis. As this picture shows, we can see in A the normal growth of ice crystals in diameter. In B, we can see the control group, how an ice crystal is supposed to grow. But now in presence of antifreeze protein 1, antifreeze protein 2, antifreeze protein 3, we can see that there is no change at all because the antifreeze proteins stop the ice recrystallization. There is no more growth of crystal. But as we can see in A20L, meaning that A20L is a defective mutant obtained from antifreeze protein 3. Alanine has been changed into leucine. And then we can see that there is now there is a growth in the ice crystal, in the size of ice crystal. That is telling us that how one single amino acid can dramatically change the role of an antifreeze proteins. Now, amino acid display in antifreeze proteins. And antifreeze proteins are macromolecules with a specific part of it that reacts, that gives the role of antifreeze proteins, meaning that some amino acids have more affinity to ice binding. Among them, we have threonine and alanine. So how to apply antifreeze protein to treat frostbite? In the cold, signals from sensory receptors are sent to the hypothalamus to start the regulation of heat. Body can keep up the regulation, therefore frostbite occur. We have three stages of it. The normal one, the first thing when the skin is red, that's the damage of epidermis. The superficial frostbite, when the tissue right underneath the skin are freeze. And then we have the deep frostbite, when the finger are black, and that means that deep the subcutaneous tissue are damaged. And when we get to that stage, there is no more chance to go back to normal. This slide is a result of frostbite experiment conducted on tails and of a transgenic mouse and the white-type mouse. In A, we can see the number of normal mouse recover after experiment. So we can see the tail that have been put in a solution at a negative 22 degrees Celsius to see what will happen. So when we had in 24 hours, we can see that the tail of the white-type mouse is red. That's the beginning of frostbite, frost nip, while the tail of the transgenic mouse is still normal. Till we get to the 7 days, up to 7 days, then we can see the damage, the complete destruction of white-type tail, while the transgenic mouse has still its tail damaged, but not fully damaged. So there is less damage compared to the white-type mouse. So in C, that is what is going. That is the inner view. That is what is going on at the cellular level. We can see that's a control group. That's what we're supposed to see. But in 24 hours, still 27 days, there's no more nucleus. There's no more blood vessels. There's no more connective tissue. So you can see that there's no more blood. Everything is white. It's completely freeze. So FPs in cryomedicine, FP are used in conservation of red blood cells. We can see here that they are using in red blood cells. The picture that is here, the slide is telling us that this in absence of antifreeze proteins, there is no more recovery. But as we increase, we can see more recovery of red blood cells. Now, what are we going to do? We would like to design a short peptide that will mimic antifreeze protein. The peptide will be synthesized and characterized to assess its ability to prevent frostbite in mice in long term. So that's what we plan to do. That molecule has been drawn using Pep Draw, and that picture here is an experiment that has done the same thing that we want to do. So what we want to do after design of this peptide above, it will first be tested for its replatization property. The successful result obtained from this experiment will facilitate escalation to mouse trial, then finally human trial. So acknowledgement in a special thanks to CSURP for the solid workshop over the program, to Professor Ryan, to my mentor Shihan, to Uline Lab Group, and to everyone involved in this program. Thank you so much for your participation, and I will be open for questions. Thank you so much.